Hello, my name is Adam Lognon. I'm a master's student at the University of Calgary in Dr. Patrick Whelan's lab. Today I will be describing aspects of my project titled The Medial Zona Incerta in A13 Nucleus as Potential Therapeutic Targets in a Model of Parkinson's Disease. The Whelan lab is interested in researching the descending neural control mechanisms of locomotion. Here I have I've depicted the canonical pathway for this descending control. Signals start in the motor cortex, which then inputs into the basal ganglia. The basal ganglia then inputs into the mesencephalic locomotor region, on into the brainstem and spinal cord, which ultimately coordinate movement. On the left-hand side, I'd like to point out the substantia nigra pars compacta. This is an important element of the basal ganglia that's crucial for its functioning due to its dopaminergic neuromodulatory input. Recently, some novel inputs to this pathway have been elucidated. One example is which has been researched in our lab, is the anatomical connectivity of the A13 dopaminergic nucleus found within the medial zona incerta, which inputs to the mesencephalic locomotor region. I'm interested in using this novel connection within a disease model. Here I'd like to present Parkinson's disease, which is a progressive neurodegenerative disorder that's highlighted by motor symptoms that include bradykinesia, which is slowness of movement, and gait dysfunction. These motor symptoms are often attributed to the loss of substantia nigra pars compacta dopaminergic neurons. My research question is, can A13 and medial zona photostimulation reduce PD-like locomotor and gait symptoms in an animal model of Parkinson's disease? I hypothesize that photostimulation will in fact reduce gait deficits in these model mice due to the connectivity with the MLR, or mesencephalic locomotor region. As an overview, I will be approaching this using a unilateral 6-hydroxydopamine regional model, which targets the dopaminergic neurons unilaterally in the substantia nigra pars compacta, ultimately resulting in gait dysfunctions that can include changes in gait metrics such as contralateral hind paw angle deviation increases. In terms of combining this with photostimulation to look at potential therapeutic effects, I will be injecting a channel rhodopsin viral construct into the A13 and stimulating within these model mice ipsilateral to the lesion. Ultimately, I will be looking at behaviors pre and post-surgery to compare for the deficits brought on by the lesion, as well as behaviors done post-surgery to look at the effects of photostimulation. Ultimately, the animals will be perfused and tissue post-hoc will be used to confirm the lesion and the optrogenic placement, or ferrule placement. One of the major features that I will be looking at to determine the effects of photostimulation comes from gait information obtained on the Digigait treadmill. In this case, mice are placed on a transparent treadmill, and that will cause them to run at specific speeds and allow for me to pull out various gait metrics, such as the paw angle. Here I have an example of a mouse running on on the Digigait treadmill, and as you can see, it tries to keep up with the speed of the treadmill, and at certain cases is able to maintain its speed where in a set location, which allows for me to obtain gait data reflective of the animal traveling at a consistent speed. To analyze this data, I will be using software developed in our lab called Gait Lab. This is built on the supervised deep learning algorithm, Deep Lab Cut, that was developed in the Mathis lab for markless annotation of images and videos. Gait Lab is a graphical user interface that allows for easy modeling and testing of models, as well as the ability to pull out gait metrics. This can be found on our Open Science Forum page with the link below. Version 2.4 has been released, and I've helped contributed with this version to different bug fixes. Here I'd like to depict the analysis page for the gait metrics. As you can see, there's a mouse on a treadmill with different annotated points. I'm circling the mouse here with the different points, including, for example, midpoints. These points allow for us to pull out the position of the paw and its phase, which allows, you can see the phase being depicted here on the bottom for the paws being in swing and stance. And then ultimately the gait metrics can be pulled out on the side here and exported for future analysis. Currently, Visual Gait Lab is still a work in progress, which is something I will be contributing to and have contributed to in an ongoing fashion. It is currently only set up for treadmill gait analysis, but we are looking to add some additional functions, including the ability to analyze catwalk data, which involves fundamentally different tracking of swing and stance phase due to the difference in the way the animal moves through the frame, as well as the ability to analyze interlimb coordination metrics, allowing us to look at how different paws interact. Lastly, I would like to try and implement a center of mass tracking approach that will allow for us to look at the position of the animals relative to the frame in the treadmill data and obtain facilitate obtaining data where the animal is in a consistent location on the treadmill. Currently, my contributions to open neuroscience include me continually working on bug fixing and testing of Visual Gait Lab, as well as feature development. I'd also like to point out my contributions in terms of running an advanced neuroscience techniques journal club that allows for the sharing of novel ideas among junior scientists and sharing open neuroscience techniques and tool development. Additionally, I've worked on a project which has been released to the OSF link below that allows for the classification of spontaneous electrophysiological data. Lastly, some of the lessons I've learned here are that collaborative multidisciplinary teams are critical for implementing new research tools. Additionally, it's important to have resources to actively communicate and share data, and timely interactions between the different members is important for progressing projects, and ultimately always always expect to find bugs. Here are my references, and I'd like to thank you for listening, and here are my acknowledgements.